Okay, so I have just received a question from a student asking me to go through the different stages of meiosis. So I wanted to cover that here. So what you should know with meiosis is that there is PMAT but twice. It's a reduction division. So these are a few of the slides from my video. So if you haven't checked out my YouTube channel, do check it out. But for those who are looking for a bit of a recap, the first thing to note is that when you are looking at meiosis, what you want to note is prophase 1, your nuclear envelope, your nuclear envelope and nucleolus will disappear. What will happen next is your chromosomes will condense, they will supercoil, and what you're saying is the nucleosomes are getting closer together, and that's the only time we actually see them visible in the cell is when they are going through the either mitotic division or meiotic division. So we have these chromosomes condensing, they're supercoiling. Then we have the centrioles move to the poles and start to produce these spindle fibers, these cytoskeleton, these proteins which are going to attach to the centromeres, and they are going to pull them apart when we get on to that point of reduction division. So homologous chromosomes then are chromosomes which have the same length with the same genes at the same location. So I've tried to represent this here where you can see that this is one of my chromosomes. I've used Play-Doh for this one, and here's another one, and they are the same length. See that this is one of my chromosomes. I've used Play-Doh for this one, and here's another one, and they are the same length. They should have the same genes at the same loci. And if we look here, we have this again where we've got longer ones, but this time I've identified positions of the genes. So here I have the gene and here I have another gene. And therefore what I'm trying to represent with this picture is the fact that you have the chromosomes which are the same length, they have those same genes at the same loci, but they can be different versions. So we've got different alleles in this case. So what are the green sections that I'm identifying? Well, the green sections on this is identifying your centromere, and it's also identifying your telomeres, which are at the end. So that is to prevent degradation of the chromosomes. They have these little bits at the end called the telomeres, and the centromere is what holds your chromatids together. So it is important when you are doing this that you note the difference between the two sections. So when we look at this then, and if I come back, what you have is as they are lining up as bivalents, which means that they are homologous chromosomes, same length, same genes at the same loci, what can happen is crossing over can occur. So that crossing over that occurs means that as they came together as bivalents, they're going to have this position where they are twisted around each other, and we call that a chiasma. So the thing for you to note is the next stage is metaphase. So metaphase 1 is where these homologous chromosomes are going to line up at the equator, which is also known as the metaphase plate. Now what's important to note is this is where independent assortment occurs. So how did they orientate themselves when it came to those homologous pairs coming together? So you can quite clearly see I have my purple and pink Play-Doh, and therefore was it a case of that it went small purple on the left hand side and small pink, long pink and small and long purple? Did it orientate that way, or did it orientate the two purples together and two pinks together, or did it orientate in which we now have crossing over? So the independent assortment that occurs, we're talking about this orientation of each homologous pair and the fact that it's independent of another. So anaphase 1 then, what are we talking about? These homologous chromosomes are going to be pulled apart. They're going to be pulled by the spindle fibers retracting into the centrioles, and that therefore is going to now separate my homologous chromosomes, so that now I have two cells with only one of the chromosomes in it. And again, we go back to that stage of metaphase 2, lining up at the equator. But this time, because there's only one chromosome made up of sister chromatids present, what's going to happen is when we get to anaphase 2, those sister chromatids are pulled to the poles. Therefore, the chromatids are pulled apart. That will then remove them from being together. What we get is that the telophase 2, the chromatids have reached the pole, are now at the point where the nucleus can reform. So meiosis is a reduction division as it produces 4 haploid cells. So let's just go through this. I'm going to do this a bit more in detail now. So here you have my cell, my parent cell, which is made up of 2N. It is a diploid cell. So the first thing we note is that it is diploid because it has 2 sets of every chromosome. Now, through DNA replication, what's going to happen is we are going to replicate that DNA because it's semi-conservative. That semi-conservative replication will occur, and what we now have is we have 2 chromosomes, but they're made up of sister chromatids. Now you should know for every single cell, if we use humans, we have got 46 chromosomes. We have 23 pairs of chromosomes. Therefore, we have a diploid cell, and there are 23. So from number 1 to 22 is our autosomes. And the 23rd pair is our sex chromosomes. So right now what we're doing is we're just making a few notes about what we know. So we have our diploid cell, 2N, we have replicated it, and now we have each of them being attached. So the sister chromatid is attached at the centromere. So here we have my centromere, which is holding my sister chromatids together. So the centromere is the one that's going to hold those sister chromatids together. They're sister chromatids because they have the same DNA sequence. So if I now bring in my blue one and I put that beside it, they should be the same length because they have the same genes at the same loci. The difference is these now, if I'm looking at a blue one with a red one, these are classed as non-sister chromatids. And that's important when we're talking about where crossing over could occur. So if we then move on to my next slide. So what am I showing here? Well, what I was asked about was what are the phases of meiosis? So if we're looking at prophase and we're looking at prophase 1, the key thing to note is that you have got the chromosomes condensing. The chromosomes condense, therefore the chromosomes are now visible. What else have we got? Well, we've also got that they will line up together, they will form bivalence. Or another way of putting that is the homologous chromosomes pair. And then what else have we got? We've got the nucleus. I can spell nucleus, might help. Disappears. and the nucleolus also disappears. So prophase 1, our chromosomes are condensing, our bivalents have formed, homologous chromosomes have paired together, our nucleus has disappeared and our nucleolus has disappeared. Well, what happens now is we've now got to go to metaphase 1. So metaphase 1 is where the bivalents line up at the equator. Or you could say the metaphase plate. So metaphase 1 is about the homologous chromosomes that have paired together being moved through to that metaphase plate, which is the center. Now, how did they do that? Now, I haven't drawn that on my picture, but what I'm going to do now is I'm going to show you the centrioles with their spindle fibers. So the spindle fibers are going to attach to the centrioles, and when they attach to the centrioles, what they will do is they will push and pull the chromosomes until they are lined up at the metaphase plate. Once they have lined up at the metaphase plate, what you're then going to have is that the centrioles will start to retract their spindle fibers. So this is where we're at anaphase 1. So anaphase 1, we have that the spindles are retracting back to the centrioles. What's that going to do? That's going to separate my homologous chromosomes. So the fact that I'm separating my homologous chromosomes, what that then means is I now have got, once they've reached the poles, I have now got only half the genetic information. They have only one of the chromosomes, but the chromosomes are made up of sister chromatids. So they are now haploid, but they are haploid cells made up of sister chromatids. So during telophase, we know that the chromosomes reach the poles. We know that the nuclear envelope can reform, and we know that what we're going to end up with is our chromosomes, which are going to uncondense. So they are now going to uncoil themselves. So then, if we move on, so what do we have now? Let me just copy this across. So we got to telophase. We have reached the poles. We have our sister chromatids, and now what needs to happen is they're going to go through the whole process again, but this time as they go through prophase 2, what's going to happen is they will supercoil again, and then we've got metaphase 2. And that metaphase 2, what's going to happen? They're going to line up at the equator. But this time, when they're lining up at the equator, what we're saying is each of these individual cells during metaphase, they're lining up, the chromosomes are lining up at the equator. So Not only did they recondense during prophase 2, the nuclear envelope disappears again. Then metaphase 2, what are they doing? They are lining up at the equator, and the same with the other one. Let me just do that one quickly. I'm going to line these up at the equator. How did they line up at the equator? Well, this is where you've got your centrioles with your spindle fibers attached to the centromeres and pushing and pulling them to that center, to the equator. Then what we're going to have is we're then going to end up with anaphase 2 happening. Now, anaphase 2 is where we're going to separate my chromatids. So therefore, what's going to happen is they are going to be pulled in opposite directions. Now, the fact that they're being pulled because of the spindle fibers attaching to the centromere, that is what is going to pull them and give the shape that we recognize as being a bit like a V. So if you're ever asked to draw them, they will expect you to draw it like a V shape. And that's because what you've got is you've got that the spindle fibers are attaching at the centromeres, and that's what's being separated and therefore pulling the sister chromatids apart. So the spindle fibers are retracting back to the centrioles, and as they retract back to the centrioles, what they then do is they will then pull those sister chromatids apart. So what you've got is them being pulled apart, and because they're being pulled apart, they're retracting back. We now will have the haploid daughter cells. So when we get to the last stage, which is the telophase, once they've reached the poles, so we go to telophase 2, once they've reached the poles, now what we have is we have one, shorter one, shorter one, shorter one, shorter one. Do this in the right colors. They have haploid because they only have one set of each of the chromosomes. So in telophase, you will have the nuclear envelope will reform around the chromosomes, and the fact that they have reached the poles is what is actually making them to the point in which they are haploid. So we had haploids, 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 and we now have 4 daughter cells which are all haploid, and they have half the genetic information. So prophase is all about that supercoiling, it's about the nuclear envelope and the nucleolus disappearing. With regards to meiosis, it's very important to note that it's during prophase 1 that the homologous chromosomes are going to come together and form those bivalents, and that is when crossing over could potentially occur. When you look at metaphase lining up at the equator, anaphase, spindle fibers retracting, they're going to pull to the poles. Therefore, if it's anaphase 1, you're going to separate your homologous chromosomes. If you're looking at the anaphase 2, then what you've got is that you are separating the what am I separating? The sister chromatids, and that then leaves you with the end point. Could you have all my slides? I am still working on the slides. Basically, I had a request today for someone asking about information on the meiosis. And what I've realized, what I'm starting to do is do these lives because I have a bit of time, and if I do it live it makes me think about it in a very specific way, and then I will put these on to the post for you later on. When you say you want all my slides, what do you want? Because obviously this is me just drawing and dotting around the stuff that I'm doing. That in a sense is just the resources that I have. And what I'm thinking about doing is maybe starting a bit of a biology club where you could get access to these of me just doing my randomness. But obviously if you want my official ones, then those ones I am in the process of building them to sell, but they're not ready yet because I want the whole course to be ready before I sell them. So I'm still working through those videos. But I do have a few questions. So I looked up a couple of questions because I thought actually I may as well jump on here and do this. So here you have 18 chromosomes in a somatic body cell of a cabbage. So if you're told a question, this is about an application one. If you've got 18 chromosomes in the somatic body cell, so straight away they've given you some information. You should know the somatic body cell would be 2N. So therefore you can make the assumption that there are 18 chromosomes in the somatic body cell of a cabbage, and that is 2N. So how many chromosomes would be present in the following? Well, the first thing you should note is you have a nucleus of the pollen grain. So the pollen grain, therefore, is the gamete. So therefore, they're checking, do you know, if you get a question like this, that this should be a haploid cell because it is a gamete, and therefore it should only have N. So if you understand that, then you can work out, well, if you started with 18, N would be 9 because you've got haploid number. Now you're asked about mitosis. So a cell that's in anaphase of mitosis. So you should know that if you've got 18 chromosomes, those 18 chromosomes are going to, if I draw this, be replicated into sister chromatids in a chromosome. So this one's a really interesting one because sometimes they will expect you to think about this in a way where if I started off with my units of DNA being 1, then I'm replicating it and now have 2, then the next bit would be where you see that you are replicating, so DNA replication. So my number of chromosomes, and this is where it's very tricky and you have to read what you're given in the exam, 18 chromosomes, therefore technically I should, even through DNA replication, have 18 chromosomes. But those 18 chromosomes are made up of sister chromatids. So in this case, they could actually expect you to do 36. So 36 if we're looking at the DNA units. However, 18 chromosomes made up of sister chromatids. It's important to note the difference between the two. And then what we have is, the next one says a cell in anaphase 1 of meiosis. So we know that our 18 chromosomes are going to be replicated and then they are going to be divided. So we start with 2N, they're going to go to N and N. Now this is at telophase 1. So during anaphase 1, in a cell in anaphase 1, I therefore know that I still would have my full set of chromosomes, which would be 2N, because they have yet to be divided. Meiosis 1, and we're talking about a cell in meiosis 1, telophase 1 will be where the nuclear envelope reforms, whereas you still need cytokinesis to separate them into two individual cells. So you have to remember that cytokinesis needs to happen as well. And that cytokinesis is where you're going to have the cell needs to happen as well. And that cytokinesis is where you're going to have the cell cytoplasm needs to happen as well. And that cytokinesis is where you're going to have the cell cytoplasm. That cytokinesis is where you're going to have the cell in anaphase 2. So because this cell is in anaphase 2, what that's telling me is that we have already reduced down to a haploid cell, but we haven't yet divided it. Now, if we're going on our DNA units, we could therefore say that there are 18 in each of the cells, or we could actually say that there's 9 that are made up of sister chromatids. So it is important that when you're going through this that you're noting the difference between what they're asking. And then this last one, a nucleus, which is in telophase 2 of meiosis. Inside the nucleus, we know that there is 9 of the chromosomes because we have got, thank you so much for the share, by the way, a haploid cell. So it's really important when you see these types of questions that you are looking at what are they actually asking you in the question. So what they could do is, oh, one second. I did the wrong one there. They could also ask you another type of question, which is looking at application of the content. So if they were to ask you a different type of question, which would be like this. So you were told in the last question that you had your cabbage and your cabbage had 18 chromosomes in it. You were told that the diploid nucleus of a hybrid offspring produced by crossing a cabbage with a radish. Now, if they're going to give you information, they will tell you that, for example, the radish also has 18 chromosomes. So how many chromosomes would be in a diploid nucleus of a hybrid offspring produced by crossing a cabbage with a radish? So we know that meiosis will produce haploid gametes. So therefore we know that through meiosis we're going to have our gametes that are haploid. So therefore if they're haploid they're going to have half the original diploid number. So therefore because of that what we would get is if they come together through this crossing, what we would result in is we would result in a diploid which also has 18 chromosomes. So we have a hybrid because we've taken the chromosomes from a cabbage and the chromosomes from the radish and we have mated it so that cross-pollinated so that we end up with a diploid offspring. Now what's really interesting is they could then go on and say, well, why would this diploid hybrid be infertile? And the reason being is although they've got this same number, so we know that it's got the same number of chromosomes, we can see that quite clearly, same number of chromosomes, 18 and 18, so therefore the haploid is 9 and 9. What we have to remember is during prophase 1, what happens? Homologous chromosomes pair up. They form bivalence. So if they form bivalence, What are they again? Remember, homologous chromosomes have the same genes at the same loci, and they're the same length. Well, if we're talking about a cabbage and we're talking about a radish, they're going to have different structures. Their chromosomes are not going to actually line up during meiosis. So different structures, so don't form bivalents. Therefore, meiosis cannot occur to produce gametes. And that therefore is why a hybrid would be infertile. So I did this video for the student who asked me about meiosis, and I hope anyone who has watched it has found it useful. And if you have, or if you still have some questions, please do put them in the comments box and I will try and respond as best I can.